Population ecology. Population ecology? What's that? Population ecology is a branch of ecology which studies the interactions of a population with its surroundings. What about population distribution? Population distribution shows how the organisms of the same species are able to spread out in a habitat. There are factors affecting the population distributions of plants, one of it being temperature. The optimum temperature for plant growth is between 25 Celsius to 30 Celsius. High temperatures can cause enzymes to denature. Biochemical processes in plants are disrupted, and they stunt the growth of plants, increase the rate of water evaporation by transpiration. They also slow down the rate of photosynthesis. While low temperatures reduce the activities of enzymes which slow down the biochemical reactions. The next one factor we have water. Water is needed for enzyme activities, photosynthesis, transport, and support in herbaceous plants. Dry regions such as a desert and tundra areas such as the poles have low population distributions and also low density of plants. The next factor we have is light. Light is very important for the process of photosynthesis in plants. Areas which receive little light have a lower number of plants. The next, which is pH of soil. The pH of soil is important for nutrient absorption by the roots. Soil in which the pH is either too acidic or too alkaline will cause lower absorption of nutrients by plants. The growth of plants will be disrupted due to lack of nutrients. The next factor is mineral salt content. Salt content affects the absorption of water through osmosis by the roots. High salt content in soil will cause plants to lose water through osmosis. Minerals are needed for the productions of proteins, enzymes, nucleotides, vitamins, and other compounds. As an example, phosphorus is used to form phospholipids. Other than that, we also have factors affecting the population distribution of animals, one of it being temperature. Areas which are exposed to high temperatures in a habitat are less inhabited by certain animals, such as worms and snails. Next is water. Animals are concentrated in areas that have enough water sources for drinking and cooling the body. Another factor is breeding site. A safe and suitable breeding site is needed by animals to raise their offsprings. Next is food supply. Food is important for survival because animals are heterotrophs, which depends on plants and other animals for food. Next, we have three patterns of population distribution. Clump. Clump organisms form separated groups. This condition is caused by uneven distribution. Random. Organisms are distributed in a free pattern in an area. This distribution involves species that have little interactions among individuals. And then we have uniform. Organisms are distributed all over an inhabited area. This distribution involves species in which every individual competes for limited resources in an area. Next, we have estimation of organism population size. What is population size? Population size means the number of organisms present in a population. Population density, what's that? Population density means the number of individuals of a species per unit area of a habitat. We also have quadrant sampling technique that can estimate the population size of land, plants, or animals which are inactive or move slowly. Here's a tip. During quadrant sampling, you should place a quadrant randomly and not based on an area with the most plants being studied can be found. The number of organisms in the quadrant areas is the sample which represents the whole area of study. The data obtained from all quadrants can be used to estimate the population in the area of study. The size of the quadrant depends on the types and size of organisms, the area of study, as well as the distributions and density of the plants being studied. What is a quadrant? A quadrant is a square frame structure which is made of wood, iron, or plastic. And in the quadrant, we have frame, which is the outside, and we have wire, which is the inside. Other than that, quadrant sampling technique can estimate frequency. Frequency is the probability to get an individual of any plant species in every quadrant. The formula to frequency is the number of quadrants containing studied species over total number of quadrants used multiplied with 100. Next, quadrant sampling technique can also estimate density, which means the average number of individuals in any species per unit area of study. Density formula, which is total number of individual species studied in all quadrants over total number of quadrants used multiplied with area of a quadrant. Other than that, quadrant sampling technique can also estimate coverage. Coverage, what is that? Coverage is the surface area of the soil which is covered by the shoots of the plant species. The coverage percentage is the percentage of soil surface covered by the plants. And the formula to coverage percentage is area covered by studied species in all quadrants over total number of quadrant used multiplied with area of a quadrant multiplied with 100. Other than quadrant sampling technique, we also have capture, mark, release, recapture technique. What is that? Capture, mark, release, recapture technique can estimate the population size of organisms such as foxes, sharks, snails, millipedes, insects, and butterflies, which can move freely in a studied area. This technique involves a number of steps. First step is animals under the study are captured randomly in the area of study, which is their natural habitat. The second step is the number of animals captured is recorded. Third step, every animal captured is marked by using nail, polish, paint, or Indian ink. Fourth, the marked animals are then released into the same area of study. Fifth, after a certain period of time, a second capture is done randomly in the same area of study. Lastly, the total number of animals captured the second time, both marked and unmarked, is recorded. And that's how you use the capture, mark, release, recapture technique.